So I'm very excited to welcome Dr. Jeffrey Meyer, who will be talking about translational impacts of PET neuroimaging and women's mental health, postpartum depression, and anorexia nervosa. And before we begin, I'd like to acknowledge that we live, work, play, and participate in a community on the unceded and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I am joining you from Toronto, which is covered by Toronto Purchase Treaty Number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members also live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And I encourage any of you to acknowledge the territories on which you dwell in the chat as we go along. So I just wanted to outline that we are the Women's Health Research Cluster. We have over 1,100 members, which include faculty, trainees, and community members across 44 countries. I'm very, very proud of that. We host a number of events like the seminar series that you're participating in today. And we have a blog. We have the Women's Health Interrupted podcast. Season 4 will be starting up probably in September again. This is our last seminar for the 2025-2026 season, and again, we'll be joining back up again in September, so look out for the announcement of our speakers for the next series. And become a member. It's free to join. If you want to support us, please support us. We're always looking for support. And with that, I will stop sharing so that Dr. Meyer can start sharing his screen while I read his bio to you and give you his fun fact, which is a very fun fact. So Dr. Meyer specializes in applying positron emission tomography to identify markers of biological change in the midst of major depressive episodes and common comorbid illnesses, and then translates these into novel approaches for prevention and overcoming treatment resistance. He has more than 150 peer-reviewed papers and has led over 20 papers published in, maybe you've heard of some of these journals, I don't know, JAMA Psychiatry, Lancet Psychiatry, American Journal of Psychiatry, Brain, and Proceedings on the National Academy of Science. These findings include a discovery of a new dietary supplement to prevent depressed mood in the early postpartum, which is one of the first probably papers that I knew I read of Dr. Meyer's. Now I lost my position. Discovering the first evidence for inflammation in the brain during major depressive episodes, obsessive compulsive disorder, long COVID, and traumatic brain injury with chronic symptoms. He's also established the 80% therapeutic occupancy rule for selective serotonin reuptake inhibitors. Dr. Meyer has received the AE Bennett Award from the Society of Biological Psychiatry, the Distinguished Investigator Award from Brain and Behavior Research Foundation, also known as NARSAD, the Innovations Award from the Canadian College of Neuropsychopharmacology, the Samarthji Lal Award from the Graham Boeckh Foundation, the John Dewan Prize from the Ontario Mental Health Foundation, and the Royal College Medal in Medicine for outstanding contributions to psychiatric research. So a lot of awards there. Well done. So Dr. Meyer's, a fun fact, he eats at least 2 cups of blueberries every day as part of his breakfast and has done so for over 20 years, except for vacations and conferences. You need to demand this as part of your, like rock stars, they make demands about only red Smarties or something. You need to have that in your before you say yes to a speaking engagement. Oh no, I can't. We can't hear you. Oh,
Okay, so great, no
oh, there
worries.
you are.
So I
Yeah.
just wanted to make sure I was ready to go. Okay,
Yes.
so thanks, Professor Galea, for that kind introduction. I didn't demand blueberries as compensation for this talk,
No.
but you'll see a connection to blueberries later in the talk. And I also want to thank the Women's Health Research Cluster for inviting me as well. Happy to be here today. I'll be talking about applications of positron emission tomography. And positron emission tomography is a type of imaging in which an imaging agent is administered to people, and then the scanner detects emissions from that imaging agent. It's a great strategy for imaging levels of proteins and receptors in the brain, although it takes a few years typically to develop each technique. And I'll be talking about applications towards postpartum depression and anorexia nervosa. In terms of conflicts of interest, I'm the inventor of a dietary supplement that I'll be talking about. So that's the one conflict. And likely I'll develop therapeutics to address some of the changes that we identify so far in anorexia nervosa. First, translational impact and imaging in postpartum depression. So we talked
Jeff,
about,
if you're showing us slides, we can't see them.
oh, I am showing slides.
I thought maybe
Okay. Hmm,
it's because I shared first, so now you have to share again.
okay, me. Glad you intervened there. Let's see here. I've got,
I think you just finished your conflict of interest. There you go. Not quite, but almost.
okay, so now you can see everything. And
Yes, but it's not in presenter mode. Or it's not in. There you go.
okay,
Perfect.
so there we go.
Perfect.
So talking about translational impact, monoamine oxidase A and postpartum depression. And as many would know, postpartum depression, the most common complication of childbearing, unfortunately, at a 13% prevalence. Not only is it the pain and suffering of having a depressive episode at the time of taking care of a young infant, but unfortunately, there are further lifelong events, so higher risk of future major depressive episodes, unfortunately, higher risk of suicide, associations of postpartum depression with greater difficulties in children having behavioral challenges or emotional disorders. And while the human suffering is the most important issue here, there is also financial cost, lost work, greater healthcare costs, cost for helping with children. It's estimated that, for example, in the US, it'd be about $40 billion a year. In Canada, say a tenth of that, about $4 billion a year. So many sequelae and problems of postpartum depression, and we lack widespread techniques to prevent postpartum depression. So I'll be talking about monoamine oxidase A in this first part of the talk, then the relationship to major depressive episodes, imaging and postpartum, and translation. So MAOA was first of interest clinically in studies of tuberculosis in which there was an off-target effect of isoniazid such that it inhibited MAOA and a sister protein called MAOB, and people that were in these sanatoriums and unfortunately separated for years as a result of having tuberculosis would not only have cure of their tuberculosis, but their mood and social interactions improved prominently, as shown in this picture here on the left. And people went from thinking of MAOA as an off-target effect of iproniazid to developing antidepressants that were more selective for inhibiting MAOA and MAOB, the sister protein, to MAOA inhibitor antidepressants. My take on MAOA was that perhaps we should be thinking about it beyond being a target of treatment to could it be elevated in illnesses or at-risk states for illnesses. And here in this diagram, you can see in the upper right, we have an electron microscopy of the end of a neuron, and you can see on the outer mitochondrial membranes where the MAOA is primarily located. In the bottom right, the different cell types where MAOA is more commonly found. GFAP labeled reflecting astrocytes. Neurons also express MAOA, particularly monoamine releasing neurons. And MAOA has important functions in terms of metabolizing monoamines, generating oxidative stress in the midst of doing that through hydrogen peroxide production, and influencing mitochondrial function. So effects that may be of considerable importance in mood disorders. When you're looking at imaging a protein, you'd like to know it has a reasonably high concentration in the brain, which MAOA does have in humans in gray matter. And we get an index with the scanning of the level of MAOA, and it's been shown repeatedly in brain tissue that MAOA level has high correlations with brain MAOA activity. So we use this imaging technique, 11-carbon harmine PET. It's an excellent technique. What one likes in scanning is that the imaging agent goes in the brain and comes out of the brain in the time of scanning, which you can see in the bottom right corner of the graph there. It has high affinity for the MAOA target. It has metabolites that do not cross the blood-brain barrier, so that's also important. And it tends to bind to the center of the functional pocket of MAOA, so it's a very relevant and useful imaging agent. And we applied it first in major depressive episodes. On the left on the y-axis, you see the index of the level of MAOA, and then the regions, of course, on the x-axis. And you can see in the dark that those in the midst of a depressive episode who at the time were not taking any depressant treatment did not have comorbidity. So this is really focusing on the differences between people in the midst of depressive episodes as compared to people in the midst of health. This was replicated in postmortem study by another group about 5 years later. They found 40% elevations in prefrontal cortex MAOA protein binding, similar elevations in MAOA activity. And then we went on to look at states with depressed mood and high risk for MAOA that also conceptually might be at states that might have high MAOA. So all these states tended to have depressed mood, high risk for major depressive episodes. And for example, we learned more in time that glucocorticoids can increase transcription of MAOA. So we looked at borderline personality disorder as well as repeat work in major depressive episodes to demonstrate elevations in the MAOA level, particularly in the prefrontal and anterior cingulate cortex. Also, toxins can, in cell models, sometimes create overexpression of MAOA. So we looked in withdrawal from heavy cigarette smoking and alcohol use disorder, again eliciting elevations in MAOA binding in prefrontal and anterior cingulate cortex that tended to correlate with mood state. And then also we looked in states in which estrogen levels may have declines, and I'm going to elaborate further on that point in the next few slides. So what was known around the time that we did the 2006 study was that there was a strong relationship across cell lines, rodents and macaque monkeys of an inverse relationship between changes in estrogen and MAOA activity, messenger RNA, or MAOA protein. The mechanism that's been proposed is that estrogen-related receptors constitutively promote the transcription of MAOA, and that estrogen effects on estrogen receptors decreases the level and function of estrogen-related receptors, at least transiently after administration of estrogens. Now, in the first few days postpartum, there's dramatic 100 to 1,000-fold drops in estrogens. And if you look at these plots, you can see that by about day 4 to 6, things are mostly leveling off, raising the issue, well, the timing of the scan ideally needs to be around day 4 to 6 if one is looking for changes in MAOA level in early postpartum, which was the intent of the study. Not an easy time to do a PET scan, but that's what we did. So you can see here a comparison of women day 4 to 6 postpartum as compared to women not recently pregnant, the index of MAOA level on the y-axis, and that there's differences throughout the brain regions of the gray matter regions across the two groups. And here in a voxel-based analysis, we have a similar kind of finding of differences between women day 4 to 6 postpartum and women not recently pregnant. And 40% is a dramatic level of effect in positron emission tomography kind of research. So at the time of the postpartum blues, we have some of these changes. Postpartum blues, what I mean by that is the common syndrome occurring in up to 80% of women, typically starting around day 3, peaking at day 5, composed of sad mood, crying spells, anxiety, restlessness, troubles with appetite, difficulties with energy and sleep beyond the challenges of taking care of a newborn. While unpleasant, it's considered within the healthy range. But an important clinical impact is that when the postpartum blues is severe, it's associated with at least a 4-fold greater risk of later postpartum depression. And at the time of these postpartum blues, we have this phenomenology going on of the dramatic drop in estrogens and this rise in MAOA level in regions that influence mood and the symptoms of postpartum blues. And one can think of the effects of elevated MAOA level as contributing to greater serotonin metabolism, being the primary metabolic route of serotonin, and also that it met elevations in norepinephrine and dopamine metabolism as it metabolizes these monoamines. And in the process of metabolizing these monoamines, it also creates hydrogen peroxide. So this is all going on around the time of postpartum blues. So I invented a supplement intended to counter the effects of elevated MAOA. It included tryptophan, precursor to serotonin as a strategy to counter elevation in MAOA metabolism of serotonin. Tyrosine is a precursor to dopamine and norepinephrine as a strategy to counter elevated MAOA metabolism of dopamine and noradrenaline. Also, in terms of the timing, the idea was to give tryptophan the night of day 4, tyrosine the morning of day 5. They both get transported in the brain by a similar mechanism, so they're intentionally separated in time. And then as well, there were 4 administrations of blueberry juice and extract rich in anthocyanin antioxidants given over day 3 to 5 to counter the potential of elevations in hydrogen peroxide. Now, when you're giving something to improve mental health that might be a natural health product, you'd ideally like it to be out into the brain. And so here I show that oral administration of tryptophan on the left is associated with similar levels in raising both blood serum levels and cerebral spinal fluid concentrations at doses that are comparable to what would be in the supplement that I'll be talking about. And then also, oral tyrosine has been associated with raising cerebral spinal fluid levels of tyrosine. So, as would be expected, given their transport across the blood-brain barrier, that they do actually affect their levels in cerebral spinal fluid. The anthocyanin antioxidants are sort of a mix of about 15 of them with similar chemical structures, and the one that had been looked at showed good bioavailability in humans, as shown in this graph here. There have been radio-labeled anthocyanin-containing fruits administered to rodents, and again, you can see a couple of peaks of better bioavailability for anthocyanins, and in the bottom right, you can see the rectangle indicating that about 2% of these are reaching the brain. One of the strategies in having tryptophan and tyrosine is that they're already found in breast milk, and primarily as proteins and peptides, about 99% or more are in this format, so it's unlikely that an oral dose would then affect total levels of tryptophan or tyrosine. But so we empirically demonstrated this just to prove the point. On the left, you can see the plasma levels of tryptophan after 2 grams or 4 grams going up about 6-fold. Didn't really matter whether they gave 2 grams or 4 grams from the perspective of what got into the blood, although people tolerated the 2 grams better than the 4 grams. And so that was a dose that was carried through. In regards to the breast milk, the proteins are hydrolyzed in the breast milk and overall levels of tryptophan are looked at, and you can see that they don't really change after giving oral tryptophan as would have been expected. Again, we did a similar study, but this time using tyrosine. Tyrosine levels at the 10-gram dose raised the blood plasma levels by over 6-fold. Where on the right, you can see that the overall total levels of tyrosine in breast milk are unchanged over the same time period. And then we went on to an oral open label study of the supplement. For this study, we had 40 women completing the assessment at day 5 postpartum after either getting no supplement or getting the active supplement. It was generally well tolerated. For this study, we had done a depressed mood induction, and the depressed mood induction meant listening to sad music, hearing the sad statements, and then seeing how score of depressed mood on a visual analog scale changed. And you can see on the left that in those not getting the supplement, there was a rise in the visual analog scale, whereas those on the right had no change in the visual analog scale who had received the supplement in the open trial. This was done before COVID, you can see, and finished in 2017. We went on to a randomized double-blind placebo-controlled trial with the first aim to be, again, looking at the effect of the supplement on the depressed mood induction with outcomes of visual analog scale and profile mood states. However, the reviewers of the earlier study said, look, you really should be looking at overall severity of postpartum blues, and the Stein-Materney Blues Scale was then added to the study. And then as well, the reviewers were very interested in looking at subsequent effects of depression over the following 6 months. And as I'll elaborate a little further, the study was run during COVID, so there were telephone contacts to look at levels of depression. So we used the Center for Epidemiological Studies depression scale to look at depression extending out to 6 months, so the severity of depression symptoms. So it's a similar kind of approach with the previous study, starting at the third trimester for recruitment, following out to 6 months. And here's the table to show the randomized double-blind groupings. And you can see if you go down to the bottom that we had 52 in the placebo group, 51 in the active group. A lot of the dropouts had more to do with maybe someone had COVID symptoms or a side effect related to COVID as being the more predominant issues, which then led to people coming out. The study completers ended up being 100 at the 6-month follow-up and 103 here at the postpartum blues point. Looking at the groupings, you see the similar age, whether you've got supplement or placebo. Most of the settings are or symptoms and situations, people's backgrounds are not particularly different between groups. The only thing that was different is that there were a couple of people prior to getting the supplement who happened to go into a depressive episode. And that might have to do with the stress of COVID at the time because the study was run during the COVID pandemic. Otherwise, the groups are similar, but the two with depression was prior to getting the supplement or placebo. Now, when we did the depressed mood induction, we had a couple of adaptations where initial study we had done it in our lab. This study we did in people's homes. Unfortunately, we ran into ceiling effects because in the midst of COVID, women were concerned about getting COVID. They were concerned about their babies getting COVID. They were concerned about their partners having symptoms of COVID, leading to them not coming into the place where the women were giving birth. As well, we ran into challenges with running the depressed mood induction in people's homes as there were other children and distractions that came into play. And ultimately, we found no difference with the depressed mood induction between those getting the treatment and those not. However, as recommended by the reviewers of the first study, we should be looking at overall severity of postpartum blues. And here's where we found differences between the active and the placebo. The vertical red stripes represent the median. And you can see that in the placebo group, it's around 4, whereas in the active group, it's gone down to about 2, so about half. And if you look at the bars, you can see in the active group, there's a shift towards the low grouping having the highest bar. With the quantile regression for the median, we found that the p-value is 0.06 there. If you use a general linear model with crying before the supplement as a covariate and looking at the effect of supplement, 0.008. Interestingly, the statistician has suggested that if one looks at other sorts of markers in terms of this quantile regression, you can set 50% the median, which is what I showed in the previous slide. But if one uses higher levels of quantile, which would suggest relevance to more severe blues, that the greater the quantile, the more the difference, suggesting that the supplement is more useful when someone has more severe postpartum blues versus less postpartum blues. I mentioned earlier that there were a few people that were in the supplement group that prior to getting the supplement happened to have more crying, and a few that happened to have a depressive episode symptom levels prior to getting the supplement. And so we also looked at the effects of removing those individuals. And as you would expect, it only increased the significance. So the top row represents everybody included. And lower rows are if you remove some of the subgroups of people who perhaps had, more outlying severity of symptoms prior to getting supplement or placebo. So the effects were consistent regardless of how one did this or if one keeps the full group. So this is a rating of people's symptoms. And the way we did it is that we would have a report of symptoms at day 3 and a report of symptoms at day 5. And if there was a new onset of a symptom or an elevation in a symptom, then it would become reported on this table. What was interesting is that those that got the supplement had a tendency to report having no symptoms of change between days in 3-5 at a trend level of the 0.06. Certainly would suggest that the supplement was well tolerated. It would be expected that women would have symptoms going from day 3 to 5 postpartum, so it's not surprising that there are symptoms reported. So I'm going to move on now to this SD. So this is the severity of depressive symptoms, and at pre-treatment, one would expect across both groups was similar, not surprisingly, and it stays pretty similar, but then at the 3 and 6 months, you can see a split between those at the active being lower in severity of depressive symptoms as compared to those with placebo. The next slide here details this out more carefully. So on the upper left corner, you can see the active pretreatment. They're pretty similar scores between those that have active and those that are placebo shown in the histograms. If one goes down to the placebo in the bottom right and the active, again, you can look at the comparison. And what's interesting, I think, on this graph is that you can see 16 is sort of a clinical thresholding. And in the active group, everyone is below 16, whereas in the placebo group, about 8% of the women are above 16 on the scale, which would be in the clinical range. Another potential interesting direction of use of this supplement, and this would, of course, take clinical trials and development, might be to consider looking in perimenopause. Here's an MAO imaging study that we've done in perimenopause. As you may know, that the fluctuation in estrogens can be about 5-fold greater in perimenopause. And what we found was that women at ages 41 to 51 who had concerns, particularly around crying spells in perimenopause, had higher MAOA levels in the gray matter areas of the brain, which tended to subside more in menopause. I guess the other comment I should make about perimenopause, as many may know, it's a high-risk state for getting to onset of depressive episodes, with about 1/6 of women having new onset depressive episodes in perimenopause, another 1/6 having clinically significant symptoms related to depressed mood and anhedonia. So strategies to prevent consequences of elevations in MAOA might be a strategy to look at, in future work with the supplement. So in the first part of this talk, I've identified elevations of MAOA level in the midst of depressive episodes. I've commented on how elevations in MAOA levels may occur in certain states that are associated with depressed mood and high risk for later onset or recurrence of depressive episodes. Then I identified a pathway of dramatic drops in estrogen in the early days postpartum, followed by a rather substantial elevation of over 40% in the rise in the index of MAOA level in early postpartum around the time of the postpartum blues, a major risk factor for later postpartum depression. I talked about the invention of a dietary supplement associated with raising tryptophan blood levels by over 6-fold and tyrosine levels by over 6-fold. Its assessments for safety in breast milk. I talked about an open trial in depressed mood induction and then a randomized double-blind placebo-controlled trial that had strong effects on reducing the overall severity of postpartum blues and showed longitudinally that those who got the supplement had lesser severity of depressive symptoms. And now I'll talk about the second part here. So the next part is a different extension of PET imaging, also relevant to women's health in anorexia nervosa. So for this part, I'll talk about anorexia nervosa, the case for neuroinflammation, some imaging of translocator protein in anorexia nervosa, and some implications. So anorexia nervosa is a debilitating psychiatric illness affecting 1 to 2% of adolescents and adults. Over 90% of the cases are women, which is why I thought it would be relevant to today's talk, of course. 3 key symptoms are extreme concern of gaining weight, severe body dysmorphia, meaning a misperception of the body in terms of its mass or appearance, and maladaptive weight loss oriented behaviors, so strong efforts to try to lose weight. And in this sample that I'll be showing today, everybody at some point had these symptoms. Look at that. And the but some people would not still have the persistent loss of weight. But everybody they either have a current anorexia nervosa or a partially remitted anorexia nervosa because they may not have the weight loss still. That being said, all the people that participated in the study with anorexia nervosa certainly were clinically affected, having at least several hours a day of concern around either their current mass or food intake or misperception of their body. So they were still quite affected, just not everybody had the weight loss at the time of the assessment and the scanning. So unfortunately, this illness is associated with a very high mortality rate, 5% dying within 4 years of diagnosis, and only 50% of people with anorexia nervosa respond to treatment. And we don't have any evidence-based pharmacological interventions. So we need to understand more about the neurobiology of anorexia nervosa to try to come up with strategies that may help from a pharmacological standpoint for helping with this illness. There's been advances in therapy, but on the pharmacology front, it's really been quite limited. So there's been some observations that raise the idea of a new angle for anorexia nervosa around neuroinflammation. So infections associated with going to hospitalization have been found to be associated with high risk for anorexia nervosa in a substantial cohort of over 500,000. There's been associations between recent immunization and onset of anorexia nervosa in a rather substantial cohort as well. The prevalence of autoimmune disease is higher in anorexia nervosa. And the presence of anorexia nervosa symptoms is quite common in a syndrome called pediatric autoimmune neuropsychiatric disorder associated with streptococcal infection. So this is an infection of a group A beta hemolytic streptococci that traditionally people think of as being associated with obsessive compulsive disorder. But equally relevant primary diagnosis can be either obsessive compulsive disorder or having a rather severe change in food intake restriction. And so this illness of PANDAS as the short form is also associated with anorexia nervosa, not just OCD. So we have translocated protein imaging as a strategy that we can look at, and for microglia, probably about 7 to 10% of the cells in the brain, they're usually in a more of a sensory detecting state with small cell bodies as shown in the upper left diagram. And when they're exposed to damage-associated patterns or they can sometimes then increase their cell body size, thicken their dendrites, and then they'll look like one of the bottom two diagrams there, where they have either amoeboid shape in the bottom right, or the cell body with large dendrites there on the bottom left. And they express more translocated proteins. So that became a strategy for developing imaging to detect this kind of change. What was found in animal studies is that when one provoked inflammation in microglia, they would typically have this kind of change. But in postmortem studies in humans, they noticed that both microglia and activated astroglia will sometimes have greater translocated protein binding, typically either in the midst of the transition or if they've transitioned into the different morphology that I was mentioning. Also, if one is a postmortem researcher here and you're trying to think of correlates in postmortem, IBA1 tends to correlate rather well with translocated protein binding. And so the difference between people in the midst of health and people in the midst of illness is tended to view as being attributable to changes in activated microglia and to some extent astroglia. And that's how the scans are interpreted typically. So we have another great imaging agent for translocated protein. On the bottom right, you can see that the imaging time, you can see the time activity curves there. And what's attractive about the imaging agent is that you get a rise in uptake of the imaging agent and then a decline over the time that you scan. Has high affinity. The radioactive metabolites don't cross the blood-brain barrier. And another issue that sometimes comes up is, well, what's the contribution? Because translocated protein can also be found in endothelial cells. But in general, with imaging, the vascular contribution is about 4% of your signal, and so the endothelial cells surrounding the vascular contribution will be lower. So there's not reason to think that the endothelial cell component is substantively contributing to the signal of what we're picking up here on the imaging. So main findings, strong elevations in translocator protein binding in ventral striatum, anterior cingulate cortex, and thalamus. Best interpretation of this is that it likely reflects gliosis of microglia and to some extent astroglia. And interesting that the anterior cingulate cortex correlated positively with eating concerns on multiple scales. Now you might say, well, gliosis has been shown before in other illnesses. What's so special about finding an anorexia nervosa? Well, one of the things is the magnitude. The only other illness that has anywhere near this kind of magnitude of change in the 50% is Alzheimer's disease in the hippocampus. This is also arguably the largest magnitude change ever found in the brain in anorexia nervosa. The regional distribution is different in anorexia nervosa, being in the anterior cingulate cortex, thalamus, ventral striatum being particularly high regions. And there may be opportunities to look at intervention strategies. This is an illness that's clearly in need of additional ways for cures, and we've now identified markers that are strongly suggestive of microglial and to some extent astroglial activation. Now, what we get with the markers are changes largely reflecting microglial activation, and microglia can have pathological protective roles. There is a tendency for the field to view the TSPO labeling as somewhat more likely to reflect more of the pathological than the protective, but I wouldn't say that every study is consistent in one direction versus the other, in every circumstance and experimental paradigm one versus the other. But, pathological concerns of microglial activation could include, increased cytokines that promote more inflammation, greater production of reactive oxygen species, diversion of tryptophan metabolism towards chiarinins versus serotonin, and greater synaptic pruning, which sometimes could come to excess. Whereas protective roles may be production of cytokines that can dampen some levels of immune responses, phagocytoting, removing debris, increasing synaptogenesis. But with translocated protein, we don't ultimately know that. In trying to decide whether this change is reflecting a cause, an association, facilitating or promoting illness, or a consequence, we do have these associations that when inflammation is provoked, that people have these higher rates of getting OCD. So one might think that given that these inflammation-provoking stimuli can also elevate translocator protein, that maybe this would be suggestive that this is more likely to be causal. On the right, I show the study administering lipopolysaccharide to humans provoking elevation in translocator protein. So we'd have some tendency to favor causality, and under that kind of concept, it may be useful to think about repurposing some medications already being developed to interfere with microglial activation to help with neuropsychiatric illnesses like P2X7 inhibitors have been designed to inhibit microglial proliferation. Inhibiting the pathway of the tryptophan towards the chiarinin pathway versus serotonin is another strategy that inhibitors have been described for inhibiting 2,3-indolamine dioxygenase. And also medications like minocycline may do two things. They may reduce the elevation in translocator protein levels in animal models and also shift cells to more curative roles. Now, the other approach is to consider whether using the imaging as a strategy to direct clinical trials and stratify so that one can identify people with anorexia nervosa who have more prominent changes like this and then therefore get a better signal in clinical trials. We tried some of that with celecoxib and depression, and so the idea here in the baseline severity and on the x-axis there is the level of translocator protein, whereas on the y-axis we have the change that occurred in symptoms after administration of a COX-2 inhibitor, suggesting some level of relationship. The change in symptoms wasn't quite as prominent as we would like, but it was showing the benefits of trying to use stratifying in a match. We did try minocycline as a strategy to lower translocator protein binding in the midst of depressive episodes, and unfortunately we're unable to do so. So it may be that the dose just isn't high enough in a human being, about a 20th of what you give to a rodent, and so that's probably the issue, and you'd have to come up with a way to address that. But irrespective of the details, because of the elevation in translocator protein binding being strongly supportive of greater microglial activation and to some extent astroglial activation, it certainly could be possible to look at interventions that might shift these glia to more curative roles. Since they're there and likely being active, then one can think about in the shorter run, this may be leading to different ways to cure. I think as well, we need to understand more details around the nuances of the changes in inflammation that we're detecting with the translocator protein. And so I think a combined approach is probably ideal. Perhaps with greater resolution on the new scanner that's coming, we might also try to look at resolving additional circuits that relate to appetitive functions that we can't pick up with the previous scanning. Even though it was the highest resolution scanner of its kind for the last 20 years, there's a new model coming up. So overall, I've reviewed two sets of studies. The first set of studies was around MAO imaging and translational studies leading to the dietary supplement. I talked about greater MAOA level throughout gray matter and major depressive episodes. Also that there was evidence for greater elevations of MAOA level in prefrontal anterior cortex in depressed mood states with high risk for onset or recurrence of depressive episodes, the pathway of drops in estrogens, rise in MAOA binding during postpartum blues, and the postpartum blues high risk factor for postpartum depression, and the invention of the dietary supplement and its effects favorably on reducing the severity of postpartum blues, and also that extending out to 6 months, women who had received the supplement had a lesser severity of depressive symptoms. I'd like to acknowledge quite a few people, in particular, and the yellow represent people who are involved in the translational imaging work that was in the postpartum direction, particularly Julia Sasher on the imaging front, Yekta Dowladi, who did the breast milk studies and open clinical trial work. And then on the direction of the anorexia nervosa, I'd like to particularly acknowledge Mariel Lepra, who recently finished her PhD and had worked on and led the study in anorexia nervosa. So I'll stop here and I'm well positioned to take some questions. Thanks.
Yes, you are. Thank you so much. Yes, thank you. Please give your little emojis because it's so hard to give virtual talks. We can't hear anybody unless you unmute yourself and say something. But thank you so much for those emojis. Thank you so much, Dr. Meyer. That was really excellent. I wrote down 5 questions. But it's not about me asking questions. Of course, please feel free to put your hand up or put a question in the chat if you want to, or unmute yourself and ask a question. And I'm going to start asking questions. I'm going to try and keep my eye on the chat, but ask questions. So I'm going to ask one from I had some from at the beginning, but I'm going to ask one at the end there. I think you showed us a little bit of data on monocycline in major depressive disorders and not showing a difference in symptoms, right?
Oh, so I should clarify. That was the main focus of the study was to look at the effect on translocator protein binding.
Yeah, and you didn't see an effect.
There wasn't an effect on translocator protein binding. That being said, and we based that on the rodent studies, but the rodent studies per body mass were probably giving 10 to 20 times greater dose. We used the highest well-tolerated dose that a person would take. So it would be the one that a teenager might take to get rid of their acne or what have you. So,
yeah. And so I'm just wondering if you look was that across both males and females? Like, did you look at any sex specific effects? And then I have a follow-up question.
Okay. So, I mean, it was 12 who got the minocycline, 9 with placebo. Yeah. So, the power for sex effects was low. I more put that in there just to show the viability of doing that approach, and also that minocycline wasn't the way that we were going to go in the longer run. I mean, one could pursue it by looking at some strategy to raise the level of minocycline in the brain, perhaps, and despite the same dose, and get more creative. I think there's going to be a talk on Wednesday, I think at CAMH, someone who takes expertise in trying to promote extra levels of transporting medications into brains. So there could be different ways to do it than
in the way No, I was just thinking if there. I mean, I'm mixing things up a little bit, but the increase in TSPO seems to be under depressive episodes as well. Is that not true, or do we know that
Yeah,
yet?
so in depressive episodes, it's generalized.
Yeah,
It's not as high magnitude.
yeah,
We did match the anorexia nervosa. In the anorexia nervosa group, there were
yeah,
two who had a depressive episode, and we did do a match of two with depressive episodes in the control to line
yeah,
up.
yeah.
And we also did it with a full complement of healthy in the actual paper.
Mm-hmm,
And either way, it doesn't
mm-hmm,
affect the overall results. So we don't think that the difference is attributable to major depressive episodes, but it might be that because anorexia nervosa with major depressive episodes is more treatment resistant on both fronts, that
mm-hmm.
if
Yeah,
you have two inflammatory processes, even if they're not identical, they're probably spilling cytokines to each other and promoting
mm-hmm.
them and making it more difficult to get rid of
Right,
one
to figure
another.
it. Right, exactly. Well, I was also thinking about when you actually started the talking about the sort of dramatic fluctuation across a postpartum period and estrogens particularly. And so I was also thinking, look, with anorexia nervosa, you also have, I don't know the area as well, but often people become amenorrheic. So suggesting that their estrogen levels will also be low. So I wonder if when you don't see as strong of an effect, if you can stratify by, which would be, we get lower and lower and lower end, but whether or not you see any sort of signal in terms of lower estrogen levels and more changes in TSPO or
Yeah, that's a good question. I don't think we didn't focus in that direction in the biomarkers.
yeah,
We did look at 19, but
yeah,
we didn't
I know.
choose that
Yeah.
direction.
Hey,
We
it's
still
pretty
have the
good,
samples, and
19,
we can we can we
Yeah.
can look at that,
Yeah.
and it could provoke
Yeah.
future study as well, and
Yeah.
in some other directions too.
Yeah.
Um, yeah, whether that could be a dampener of translocator protein potentially, or the like might be a strategy too.
Yeah.
Yeah,
Yeah.
there wasn't an association with body mass index.
Hmm. Right.
So, so that would be suggestive that it's independent of, it's not purely it was not related to starvation effects on its own.
Right. Right. And did you look? Oh, thank you. I was just going to say, people don't let me only ask questions. So Bonnie were you able to, Bonnie, did you want to ask your question? Um, yes, there you go. So it's not just me
Hi.
talking.
All right.
Hi. Thank you for the talk. I just had a quick question. I'm sorry if I missed this, but did you assess TSPO in the periphery in addition to the regions of interest in the brain?
We didn't. I mean, it's a little tricky because you need to get it off. We probably would want it off the cells, and so uh, we didn't. I mean, the main reason to look in the brain is that the those particular glia happen to overexpress it and in the activated states. I suppose it certainly could be feasible if one preserves the cells to do that. It's a little harder because you probably would have to do your assays around the time of the scan, where we do have the luxury of batching the
Right.
existing protocols for the markers we looked at. So it's certainly feasible. It could be interesting. It's an interesting point. The other thing, it might be another way to get at that, is that the next scanner is a whole body scanner. So at least we could look at
Hmm.
TSPO in
Hmm.
periphery at the same time as in brain, depending on how far we extend down the body. So it's an interesting direction.
Great. Thank you.
Sure.
I was really struck by what you were saying in terms of like the for anorexia nervosa, the signal being, what was it, like a 40% change, I think, and the only
50,
other
50. Yes.
50%, sorry.
Yeah, that's
I wrote
okay.
down 40, sorry, 50%
Sure.
change. Um, and the only other sort of large fluctuation that is seen generally in the literature is Alzheimer's disease. And then I wondered, is it known? Is there any link between anorexia nervosa and Alzheimer's disease? Because both of them are more female.
Yeah,
There's one.
I think there's a
I
I
don't know.
think
I
there's a
don't know.
higher risk of cognitive decline, but I don't remember offhand whether there was actually risk of Alzheimer's. But we did look at this. I looked at this one point. So I
Hmm.
think in general, most of the illnesses that have some level of gliosis have some level of risk for cognitive decline. Is a, and that probably fits with the hastening model of gliosis and yeah.
Yeah. Yeah, no, it's really, really interesting. You brought up so many themes that I want to talk about, but I would like to also add space for other people to ask questions. But I'm going to flip to the Blues Away. I think that's what it's called. Yes,
Yeah, yeah,
yeah. Um, so is it available in Canada? It
We have well, we're in the process.
yeah.
The challenge is like, there's a few challenges I think in going to Canada. One is that there's categories, either you are a supplement, and you can have, no bearing on health illnesses, or you're a drug and you have to pay the drug fees
Yeah.
to talk with them. So we can have one conversation with them before they slap big price points on with them. So and the challenge is that uh, in so I think about the 1990s, there was a company in Japan that introduced an impurity into making their tryptophan,
Mm.
and then it has been since resolved as being related to the impurity. And so some people would have an adverse reaction if they took the supplement, or the tryptophan from this one company. So most of the people affected were in the US. The US has taken the stance that that was from the one company
Mhm.
in Japan. Uh, Britain just said, well, there was this episode of during the time of the 1990s, so we'll just regulate tryptophan for natural health products more stringently with lower amounts of being allowed. Canada followed Britain, and that was 14 years ago. There aren't any reports in the last 14 years of the
Wow.
syndrome occurring. So our
But we still
approach
have it.
will be to discuss with them that,
Yeah. Maybe it's time
it's never
to.
been there with one dose first, and it's never been an issue with that one company being involved with manufacturing it pretty much. So hopefully we can update regulations, but
Yes, that's what I was going to
yeah.
ask you, like the sort of pain points in terms of US versus Canadian regulatory routes
Yeah. So
and
the
just
reason
like
it's available in the US is because the
yeah,
US, even though that's where the vast majority of the
had
cases
them.
have been
Yeah.
reported, had made that
Yeah.
conclusion that it was unrelated to tryptophan.
Yeah.
So that's why it started there. And so there's a conversation planned with this. It takes time,
Good.
and
Takes time. Yeah.
yeah, yeah,
Well, good. We wish you luck with that.
thanks.
We do need our Canadian regulatory institutes to, and the US and the British and all over the world to be sort of, more up to date, perhaps in their rules and guidelines. Also, in terms of we don't have very much time, but this is an easy question. I think the anorexia stuff that you were talking about, obviously really interesting, really new and novel. It's really exciting, and given the fact that inflammation does seem to be playing more of a role and people are recognizing it more in all psychiatric illnesses, it's very exciting, right? Well, of course, it makes sense that it might be involved in anorexia nervosa. Did you? I know that 90% of the participants were female, but I'm also wondering if you did a sensitivity analysis. Did you drop the males and see if it was a larger signal? And because there's some suggestion that the immune inflammatory component could be stronger in females than it is in males.
Right.
No.
I can remember thinking that the two males looked
Yeah.
pretty comparable to
Yeah.
the females. So
Yeah.
that
Yeah.
I recall.
Yeah.
I don't think we pursued it beyond that because
Yeah. They
there
look
were
the same. Yeah. Yeah.
striking
Yeah.
differences. I mean,
Yeah.
other autoimmune disease, I think of like multiple sclerosis is
Of
sort of, it's
course.
got the sex difference, and
Yeah.
the geographic distribution is sort of similar between multiple sclerosis and anorexia nervosa as well,
Yeah.
which is an interesting
No, I didn't know that.
point.
Yeah.
Yeah,
Yeah.
in
Yeah.
terms of northern latitudes and
Oh, I didn't know that.
yeah.
Yeah. Huh.
So um, yes, I don't know if it completely matches, but there's
Yeah.
some
But
quite
there's some.
similarity.
Yeah.
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 So there's more to be understood there for sure. Yeah.
Yeah, yeah, no, I think that this is really sort of, obviously very new and novel findings. So thank you so much for sharing them with us. And with that, since we have 1 minute and soon no more minutes left, I just want to thank Dr. Meyer again. If you could give another round of applause, that'd be really great. Thank you so much. Thank you so much for your time, and thank you to everyone for joining us. please take a look at our new season that'll be coming out soon, and have a great rest of your day, everyone.